In this lecture, we will investigate the mechanism of protein synthesis, which is called translation. Like DNA replication and RNA transcription, protein synthesis can be divided into separate phases for initiation, elongation, and termination. These three stages require an assortment of accessory proteins that bind to transfer RNA and to the ribosome in order to promote the speed and accuracy of translation. Protein synthesis begins with the joining of the ribosome to the messenger RNA. The highlighted region is our messenger RNA. And then we have the two subunits of the ribosome binding together, holding the messenger RNA in the middle. Within the ribosome, there will be three transfer RNA binding sites. At each site, the transfer RNA is in contact with both the 30S subunit, which holds the messenger RNA being translated, and the 50S subunit. The aminoacyl, or A, site binds the incoming transfer RNA. Then you'll have a shift to the P site, which is called the peptidyl site. This is going to bind the transfer RNA with the growing peptide chain. Finally, it moves to the exit, or the E site, which binds the uncharged, or now the transfer RNA without the amino acid attached to it, before it leaves the ribosome. Zooming in on the ribosome, we can see that the acceptor end of the transfer RNAs in both the A and the P sites are near one another. There's also a channel that connects the site to the back of the ribosome through which the peptide exits the ribosome during synthesis. And right here is that channel where the growing peptide chain can exit. In both prokaryotes and eukaryotes, protein synthesis begins at a messenger RNA AUG codon. This sequence is only recognized by one transfer RNA that has been charged with methionine. This transfer RNA does not recognize other methionine codons that occur elsewhere in the coding sequence of messenger RNA. Therefore, AUG is a very specific codon that signals the start of protein synthesis. In prokaryotic messenger RNAs, this initiation codon lies about 10 bases downstream of a conserved messenger RNA sequence called a Shine-Dalgarno sequence. This sequence is often rich in A and G nucleotides. This sequence of base pairs with a complementary sequence at the 3' end of the 16S RNA component of the ribosome subunit. Thereby, it positions the initiation codon in the ribosome. Looking at the image, here's our 16S rRNA, and that is going to do the base pairing with our sequence in the Shine-Dalgarno sequence, and that positions everything in the proper orientation to begin initiation. And notice that our start codon is positioned in the middle chamber, which is our P site. On the previous slide, we mentioned that the methionine transfer RNA is the initiator. But that specific transfer RNA molecule is only used to start protein synthesis, and it's not used for other methionine codons. What makes this initiator unique? Well, in bacteria, the methionine of the initiator transfer RNA is modified by the transfer of a formal group from tetrahydrofolate. The resulting amino acyl group is designated F-methionine, where the F stands for the addition of the formal group. And the initiator then is known as the formyl methionine transfer RNA. The addition of this formyl group to the nitrogen atom of the amino acid residue prevents it from participating in a peptide bond. And that is key. We are trying to start the formation of a peptide bond. We don't want this to be adding on to other peptides. So this would be a good starting sequence because we are capping the N-terminus of the polypeptide. Therefore, nothing can grow on the N-terminus. We are only then able to add to the C-terminus of our polypeptide, which is how we want our peptide to grow, from N-terminus to C-terminus. After the peptide formation begins, then the entire formyl methionine residue will get removed. And that occurs in some post-translation modifications. Other than finding the Shine-Dalgarno sequence, there are other things that are going to help initiation. Bacterial initiation requires 3 initiation factors called initiation factor 1, initiation factor 2, and initiation factor 3, all abbreviated IF 1, 2, and 3. These initiation factors bind to the small ribosomal subunit to promote the dissociation of the large and small subunits. The formyl methionine transfer RNA then is going to bind to the 30S subunit with the assistance of initiation factor 2, which is a GTP binding protein. Initiation factor 1 blocks the A site of the small subunit, thereby forcing the initiator transfer RNA into the P site. After the 30S messenger RNA transfer RNA formyl methionine complex has assembled, the IF2 hydrolyzes its bound GTP to form GTP and inorganic phosphate. This allows initiation factors to dissociate and the 50S and 30S subunits to assemble. The 70S ribosome is now primed with the charged formyl methionine transfer RNA at the P site, and is ready to bind a second amino acyl transfer RNA to form the first peptide bond. Therefore, initiation does not begin in the A site. Initiation begins in the P site. Then after this, all subsequent transfer RNAs are going to come into the A site first, and then they'll move into the next sites. So initiation is now complete with the formyl methionine in the P site of the ribosome. Protein growth or elongation occurs when an amino acyl transfer RNA enters the A site of the ribosome. As I said just a slide ago, it is the initiator transfer RNA that's the only one that enters the P site without first binding to the A site. The amino acyl transfer RNAs are delivered to the ribosome in a complex with a GTP binding elongation factor known as elongation factor TU. EFTU binds with all 20 types of amino acyl transfer RNAs. And the highlighted region right here is your bound guanine nucleotide. With it binding to all 20 types of amino acyl transfer RNAs, this must mean that there is a common recognition element in the structure of transfer RNA. The recognition site is found in the T psi C loop of transfer RNA. Elongation factor TU delivers the appropriate amino acyl transfer RNA as specified by the codon to the A site. If the anticodon pairs with the codon, GTP is then hydrolyzed, and the EFTU GDP complex departs with the A site now occupied by the correct amino acyl transfer RNA. The elongation factor TS induces release of GTP from elongation factor TU, which is replaced by GTP, and another cycle can begin over again. Essentially, this is the mechanism for growing your peptide chain. It's the way that we are able to bring amino acyl transfer RNAs into the A site. Now that we have a amino acyl transfer RNA in the P site, and there is a amino acyl transfer RNA in the A site, these two neighbors can start to interact and form your peptide bond. To begin, we have the A site containing an amino acyl transfer RNA, and the P site contains a formyl methionine transfer RNA. Peptidyl transferase, found in the 50S subunit, catalyzes a transpeptidation reaction in which the amino nitrogen of the amino acyl group in the A site nucleophilically attacks the ester bond that links the peptidyl group to the P site transfer RNA. With the attack on the carbon atom, the electrons from the bond move to the oxygen atom in the adenine. That now creates your new peptide bond. And this reaction lengthens the peptidyl group by one amino acid at its C terminal end. Thus, the peptide grows in the N to C direction. No additional source of free energy is required because the free energy needed for the breakage of the ester bond is comparable to the free energy released from the formation of the peptide bond. Essentially, we have a net neutral type of reaction coming on here, so there's no use for ATP hydrolysis in this case. Upon the peptide bond formation, the growing peptide chain is in the P site of the 50S subunit, while attached to the transfer RNA in the A site. The transfer RNA in the P site of the 30S subunit is uncharged, meaning it's lost the amino acid. Elongation factor G, also called the translocase, uses energy of GTP hydrolysis to translocate the messenger RNA by one codon. Upon translocation, the peptidyl transfer RNA is fully in the P site, the A site becomes vacant, and the uncharged transfer RNA is in the E site, which now allows it to become disengaged from the messenger RNA. So here is a scheme of the reaction taking place. We have our formyl methionine transfer RNA initiating in the P site. The A site is empty. We then have binding of the amino acyl transfer RNA into the A site. Through GTP hydrolysis, the elongation factor is released, thus allowing for peptidyl transferase activity. We have interaction between the peptidyl groups, and we start to form our peptide bond. The peptide bond is now transferred to the transfer RNA in the A site, leaving the transfer RNA in the P site uncharged or empty. And then through GTP hydrolysis, we are going to shift the ribosome down the messenger RNA, moving towards the 3' end, which now puts the transfer RNAs in the new site. The A site is empty. The peptidyl site now contains the charged transfer RNA, and then the E site contains the uncharged transfer RNA. Finally, the transfer RNA in the E site is released, and we can repeat the process. A new amino acyl transfer RNA can enter the A site, and the process goes on. This process of elongation continues with the growing chain exiting the ribosome through the channel in the 50S subunit until a stop codon is reached. The stop codons are UAA, UGA, or UAG. These codons would appear in the A site, and they would stop the production of the peptide chain. These are stop codons because they are not recognized by any transfer RNA, but they are recognized by proteins called release factors. Release factor recognize stop codons and facilitate the attack of a water molecule on the ester linkage between the polypeptide chain and the transfer RNA in the P site. This hydrolysis releases the complete protein. Upon binding of our release factor, hydrolysis between the polypeptide and the transfer RNA occurs. Therefore, we cleave the polypeptide, and then we have release of the RF factor, the transfer RNAs, and then your subunits will split apart. And this splitting apart is associated or facilitated by GTP hydrolysis. As you can see, the release factor does contain a GTP component, so upon release of that, it provides the free energy necessary to break apart all of these components and terminate the production of the polypeptide. Of course, there's going to be some differences between prokaryotic and eukaryotic cells. The basic plan is the same for all organisms. But eukaryotic protein synthesis is more complex in a number of ways. The ribosomes are larger, consisting of a 40S and a 60S subunit that form the 80S ribosome. Protein synthesis begins with a methionine rather than a 4-methyl methionine. Which means a special initiator transfer RNA called methionine transfer RNA I is required, the I symbolizing initiation. And then the initiator codon is always the first AUG from the 5' end of the messenger RNA. Also, more protein initiation factors are required. The messenger RNA is circular because of interactions between proteins that bind the 5' cap and those that bind the poly A tail. Elongation and termination are similar in eukaryotes and bacteria, except that bacteria have 2 release factors while eukaryotes have only 1. Protein synthesis occurs in the cytoplasm, while RNA synthesis occurs in the nucleus in eukaryotes. And moreover, protein synthesis machinery is organized into larger complexes that are associated with the cytoplasm or the cytoskeleton. In eukaryotes, protein sorting or protein targeting is the process of directing proteins to distinct organelles such as the nucleus, mitochondria, and endoplasmic reticulum. 2 pathways are used to sort proteins. In one, completed proteins are delivered to the target. In the other pathway, called the secretory pathway, proteins are inserted into the endoplasmic reticulum membrane co-translationally. Protein synthesis in the secretory pathway occurs on ribosomes bound to the endoplasmic reticulum. Endoplasmic reticulum with ribosomes bound is called the rough endoplasmic reticulum. The synthesis of proteins bound for the secretory pathway begins on ribosomes that are free in the cytoplasm. Once a portion of the nascent protein that contains a specific signal emerges from the ribosome, synthesis is halted and the ribosome complex is directed to the endoplasmic reticulum. Once bound to the endoplasmic reticulum, protein synthesis is reactivated, with the nascent protein now directed through the membrane of the endoplasmic reticulum. Several components are required for the co-translational insertion of proteins into the endoplasmic reticulum. The signal sequence is a sequence of 9 to 12 hydrophobic amino acids arranged in an alpha helix often located at the N-terminal region of the primary structure. That identifies the nascent protein as one that must cross the endoplasmic reticulum. A signal peptidase in the lumen of the endoplasmic reticulum may remove the signal sequence. The signal recognition particle, SRP, which is a GTP binding ribonucleoprotein with GPTase activity, binds the signal sequence as it exits the ribosome. SRP binding halts protein synthesis and directs the complex to the endoplasmic reticulum. The SRP receptor, which is an integral membrane protein with GTPase activity, binds to the SRP ribosome complex. A translocon, a protein conducting channel, accepts the ribosome from the SRP receptor complex, and protein synthesis begins again with the protein now passing through the membrane in the translocon. Upon GTP hydrolysis, the SRP and SRP receptor dissociate and begin another cycle. Essentially, looking through the scheme, we have our protein being generated in the cytosol, but then we need to start moving this protein into the endoplasmic reticulum, which is going to be using the translocon, and we need to have a specific recognition site that allows the protein to move through the channel, and then as the protein gets constructed, it ends up being produced in the endoplasmic reticulum until synthesis is halted, and then we can do the protein sorting that is needed in the endoplasmic reticulum.